Um, Afternoon, everybody. I'm Steve Johnby. I chair the Department of Medical Ethics and Health Policy here at Penn, and a significant part of my own research is in the areas of ethical, legal, and social implications of genomics, including some of the issues that you all are talking about. So I'm really looking forward to being a part of the afternoon's conversation. I'm sorry that I had to miss the morning for other commitments, and I'm in particular really looking forward to the two talks that we have coming up. As you know, of course, the full buyers are in the program, so pretty good list. Talk is by Shawneequa Collier, who is associate professor at George Washington University, collaborating globally and equitably to distinguish ancestries from racial categories in genomics research and medicine. So, Collier, welcome. Thank you.
Good afternoon. It's a pleasure to be here. Thank you to Dr. Roth and other organizers for me. I'll start by saying this title refers to African diaspora populations, and I chose to highlight African ancestries because most of my work focuses on the African diaspora, but a lot of what I'm going to say would apply to a lot of the broad ancestry groups that we use. And I also want to preface this by saying that I understand and believe in the importance of social race and cultural aspects of race, but what I want to talk about today is biology and research into biology and understanding different ancestors in medicine and how ancestry influences medical care. Is there a way to move? Right. So to start, I don't have any conflicts of interest, and the opinions expressed are my own. Here is my agenda for the discussion this afternoon. Given the focus today on genetic ancestry and perceptions of race, I will discuss some of the barriers to understanding genetic ancestry and how we render diverse genetic ancestry invisible in research and when we use social racial categories. I'll be speaking from my lens as an ELSI researcher. I will discuss the concern that the path to understanding global genomic diversity has been narrow, uneven, and uncertain. We sometimes look to data collection and better technology to solve this problem. We can just collect more of our samples. If we can just improve our genomic sequencing technologies, we won't get over this hurdle. But these are not enough. We have to widen the path beyond convenient samples and convenient relationships and innovate how we partner. And so I want to combine today discussion about including more people in genetic ancestries and research, and also thinking about the broader community of researchers who are included, the scientists that are included from around the globe. So first, I'll start with our ethical responsibility to widen this path. As most of you in this room know, the National Commission for the Protection of Human Subjects, Biomedical Behavioral Research, authors of the Belmont Report, our foundational research ethics document, emphasized that investigators should ensure that those we recruit to research are not routinely the same groups who are excluded from the benefits of research. There should be some benefit for those who we recruit to research. The commission also argued that the products of research should not advantage only those who can afford them. International guidelines go further by acknowledging community and country health priorities, needs, and access to benefits. Based on these few examples that I'll list here, I won't read them due to time, but you can see that the language from the international frameworks I've quoted talk about community benefits and addressing the priorities and health needs of communities. So based on these few example guidelines, we can argue that there are some ethics frameworks that urge the scientific community to consider country and community health needs, priorities, and access to benefits. But who is included in the definition of scientific community? Are we including the voices of underrepresented scientists? This is an article I co-authored with geneticists Drs. Bentley and Rotimi at the National Human Genome Research Institute. Consistent with our work predominantly focused on African ancestry populations, we evaluated a number of years ago some of the successes of genomics research programs that are including increasing numbers of populations with African ancestries. You can see in this chart that we listed a number of consortia and research programs. In NIH's All of Us program, at the time of our publications in 2020, over half of the samples and data came from underrepresented populations. And you can see in this figure here that at the time of publication, it also had the greatest number of samples from African diaspora populations. Among the GWAS catalog at the very end, however, only 2.4% of published GWAS focused on populations with African ancestries. What stands out is that although these studies only accounted for a relatively small percentage of published GWAS, they were able to provide unexpectedly high levels of insights into how we interpret genomic variation for all populations, African ancestry populations, but we are able to talk about what we're seeing in European populations better because we're including people from diverse ancestries and from African ancestry backgrounds. A benefit we acknowledge here is the potential to better understand global genomic diversity for all of humanity. Scientifically, we need diverse populations to benefit us all. But how can we move away from race, right? That's the conversation we're here to talk about today. So as I will discuss next, I believe that there's a disconnect. If we're not able to encourage and facilitate discovery in diverse ancestry groups, we're going to continue to have the problem of using really broad categories to talk about our research populations. And I will draw on some of the arguments that my colleagues and I made in this article about barriers to studying diverse ancestry groups. So what are some of the underlying reasons why we haven't moved forward in terms of including more diverse ancestry groups in our work? To get to this point, we can start talking about genetic ancestry. So to start in the clinic. We talked about lagging diversity in the article. Here's a US example that was published more recently. The American Society for Human Genetics published a workforce diversity report in 2022 based on survey data from individuals in various genomics fields, including geneticists and genetic counselors. The respondents' self-reported race reflected very low percentages of representation from minoritized groups in the United States. One of the arguments my colleagues and I made in 2017 is that the fewer the scientists with diverse backgrounds and perspectives, the more likely the field will miss out on hypotheses and priorities that experts from different communities and groups would bring, whether in the United States or globally. Thinking about this on an international level, there could be opportunity for significant and sustained public health research to be conducted, for instance, on neglected diseases when research is led by investigators in different settings. But as we know, priorities often center around those with resources. So another overarching concern is that when we do partner and bring in other communities, that there are inequities in these relationships. And so I want to start by giving an example that we may all be familiar with. And Dr. Janet Shim and Dr. Sandra Sujin Lee and I were on a project where we're studying research partnerships and what makes them equitable. And so we've been thinking a lot about these kinds of relationships. It's called the Justice Project. So typically researchers, you might hear a researcher say, well, if you're an investigator in a low resource setting, you should send that sample to a high income setting. That sample and data should be exported to high resource institutions where you can centralize the analysis, where the analysis will be more efficient, where the investigators can utilize state-of-the-art equipment, rely on their concentrated expertise, standardize the quality testing, provide high quality laboratory infrastructure, and I could go on, right? And so for a long time, we've had these kinds of relationships where we believe it's okay to export samples, but then there are challenges to that actual partnership. Even if the collaboration starts off equitably, African researchers who may be operating in low resource settings could be marginalized later when the funding runs out for them, but not for their collaborators. What will happen to research samples, data, and research projects when the funding ends? Local researchers may want to drive their own research agenda. Here we get back to the question of the agenda. There may be opportunities for training and skill development. So a second problem after lagging diversity is inequity in the partnerships that we have been building. Another factor is this idea of the preferred cohort. We called it the preferred cohort effect. And then after that, flawed comparisons and expectations. European ancestries, the disproportionate representation of European ancestries reduce and the reduction of interest in other ancestry groups, the lack of studying genomic variation in other ancestry groups. So European ancestries, we argued, have been the preferred cohort for study and publications because there are incentives for researchers to prioritize European ancestry populations. Since genome scientists have traditionally focused on studying populations of European ancestry, the field has accumulated large data sets ripe for research on European ancestries. When researchers submit their work for publication or for funding, they must often demonstrate to the peer reviewers that they have a large enough sample size for their study to be considered impactful. Consequently, researchers often choose to build upon existing data sets that contain mostly European ancestries or data from European ancestry populations. And they do so to demonstrate that they have found something novel in a very large data set. As researchers are under pressure to publish frequently, they may choose existing, easier, convenient population data sets rather than engage communities or continue to do the work or ongoing community engagement that's necessary for other diverse populations. They may also avoid collaborating with investigators in countries with strict data protection laws and policies that require, for instance, that maybe the local investigator serve as a co-leader or that samples or data stay within the country. The result is that the inclusion of samples from European populations has been widely successful. Second is the concern of flawed comparisons. Journal peer reviewers looking for high impact may hold investigations into underrepresented populations to the same standard as they would for studies in European populations, expecting a similarly large sample size, even if the findings are scientifically valid. In addition, they may treat European ancestry cohorts as the standard against which other ancestry groups are compared, leading to constant continental or racial comparisons about who is represented and what genomic findings are found. It may be harder to publish findings when new variants are found in underrepresented populations and smaller sample sizes. And so dramatic evidence of this is the genome, the GWAS diversity monitor. This snapshot was taken a month ago, April 10th, 2020, so almost a month ago. And you can see here the low amount of representation of underrepresented, marginalized populations in published GWAS studies. This Nature article also reports another disparity. Although investigators may have a sufficient sample size from diverse populations to conduct scientifically valid analyses, investigators commonly discard these data or use them only to replicate or confirm findings from studies focused on European populations. The Nature article notes that aside from sample size, there are many reasons why investigators justify their focus on only European ancestries. And I'm going to say this in terms rather than how a geneticist might say it. The data sets for European populations may be more efficient to study given current understanding about population structure around the globe. Investigators may exclude the presence of genetic variants common outside of European ancestries in order to prevent errors in their analyses. The result is that investigators often summarize data from underrepresented populations only as a comment on the generalizability of the primary results, or they may choose not to summarize the results or choose to summarize the results only in the supplementary text. So aside from the ethics of recruiting people to research and then not considering the data equitably, data from underrepresented populations are placed on the periphery. These may be racialized others. And if we don't study these groups closely, they continue to run the risk of remaining homogenized. We have to treat these data as, and I'm arguing this, as valuable in and of themselves, to do a deep dive into these diverse ancestries that we're recruiting to research. It's also important to invest in technology. How can we ensure that we're able to sequence and analyze samples from diverse populations with high quality accuracy? And it's not that populations are so very different, but it's just the amount of diversity around the globe genomically, and having the tools to go beyond what we're used to in a European setting, and being able to sequence, analyze, and understanding what we are seeing in other countries due to the changes in population structure, the differences. So for a long time, many commercially developed genotyping chips were developed using DNA samples from limited populations. And so they did not work well on samples collected in Africa. The chips used for genetic identification can be less efficient in identifying the many common genetic variants in Africa that exist in other parts of the world, but are less frequent in other parts of the world. As an illustration of these clinical consequences, consider that a few years ago, Mayo Clinic researchers reported that Black participants in the Cancer Genome Atlas received lower quality genomic sequencing, leading to incomplete data. The Cancer Genome Atlas is a publicly funded project that catalogs and identifies significant genetic alterations responsible for causing cancer. Examples like this show that we don't just have a problem when it comes to recruiting, but also actually analyzing and reporting the data. The Mayo Clinic author suggested that the genetic tools may not have been as effective as those used for European ancestry. I want to share some good news. So we talked about NASM earlier. There are so many genomic researchers who are deeply committed to moving beyond race and studying diverse ancestries, and also developing the tools, the technologies, and the methods so that we can go back to these databases, combine genomic variations from different ancestries, and actually do the work to understand how variation works in diverse populations. There is a commitment to this work being done, and we're seeing some investigators and consortia devoted to these projects. And I think that will help us to get closer to understanding genetic ancestry. But one of the points I also wanted to make today is there needs to be more partnership as well as we build these technologies. So before I get to that, I want to transition into the clinical implications. Our colleague earlier this morning, Deputy Director Vince Bonham, is a co-author on this paper, and he and his colleagues highlighted the perils of lack of diversity in clinical contexts. Without a doubt, the lack of evidence on how diverse ancestries affect clinical interventions will mean that only those with European ancestries will benefit, or those who have genetic variants that we understand because we've seen them in European populations. And so what would it mean clinically if I have significant ancestry from a group in Central Africa, if that group has never been a part of the genomics research study? Further, and very pertinent to our discussions today, is that broad continental ancestry categories are likely to be racialized. I think a few people made that point this morning. As MIT Technology Review reports, companies and laboratories such as Collier Genomics, Myriad Genomics, and Ambry Genetics initially marketed something called a polygenic risk test, polygenic score test, only to European individuals because the tests were developed in European ancestry populations. Researchers often warn that scores discussed in one ancestry group, i.e. European, are not transferable to others, e.g. African. Non-experts don't always understand that the estimates can be less valid, even with individuals of the same genetic ancestry. They can still be less valid in European ancestry groups if we don't account for study design, questions about age, socioeconomic status, and demographics, sex. Further, since this language makes it sound as if the score can be racialized, doctors or companies communicating with Black, Hispanic, or Asian populations may similarly refer to race as a caveat to say that the scales don't work well in the person's racial group. Ideally, we would one day develop an algorithm to treat individuals regardless of race or known broad continental ancestries. But we need to understand the global genomic diversity within continents in order to do so. And this is an example that's been around for a long time related to pharmacogenomics, and it has to do with the HIV drug, Bacavir. And initially, scientists discovered that European Americans were likely to have a genetic variation that caused a deadly reaction to a Bacavir. And so it was recommended that the FDA screen all European ancestry Americans before giving them the drug. Later, researchers looked at how those variants showed up in other population groups and found that Gujarati Indians in America actually had the highest level of variation of that particular allele. But what's also interesting here is if you compare groups that we might call Black, the Luya versus the Maasai in Kenya, groups that are right next to each other, you can see that there is a difference in the prevalence of the allele, and that those differences are very different from groups that would be labeled African Americans. We see similarly with Asian groups, Chinese Americans versus the Japanese in Tokyo versus the Chinese in Beijing, that these are different levels of prevalence of the allele, which can have significant clinical implications if we are treating all patients as a single broad group. There are social concerns as well. Will the global genomics research revolution of the future be viewed as trustworthy and equitable? As these authors have explained, we need better, more trusting relations between scientists and scientific institutions on the one hand, and their many publics on the other. And this point raises the question, are we doing enough to address the social questions around how we label people, representation, and ancestry? So let's start with the international context. I'm sure many of you in this room are familiar with Dr. Fulwali. I may be pronouncing her name wrong. I know she has a new book out that came out in April where she talks about race, but I really love this example, although it was published a while ago. And she says that here she said that she is Black in America and white in parts of Africa. In the course of a plane ride, her race could change from Black to mixed if she flies from America to Senegal to France. In the United States, many patients come from around the world and may not define themselves in the way that we define them, using our OMB census categories. Of course, depending on the doctor or the case, someone from Ethiopia, for instance, would be treated as Black, right? Or African American, even though the percentage of African heritage for any person who identifies as Black can vary. And also it changes as you move across America statistically. So this raises questions about how we are valuing populations. A concern I want to end with before I talk about new models of collaboration is that we're not paying enough attention to multi-ancestry. Of course, there are geneticists who talk about multi-ancestry and the fact that a lot of patients are genomic mosaics of their ancestors. We know that where we look on a particular part of the genome may represent an ancestry that's not aligned with how a person self-identifies, but you can't tell that by looking at the patient. But there's an assumption that if we just enroll diverse people, then there's no problem. The labs will have diverse samples and data, but who will have the voice and the power to help us talk about and distinguish these different labels that we're using? How do communities benefit from how we're defining them, from the research questions they're asking? Do diverse communities see themselves in the labels that we're using? So you have a lab of scientists that's recruiting from a diverse part of the world. when they report their results, will people feel as if they've been represented? Will they feel as if the labels align with them? These are the questions we also should ask as we develop these modern technologies and methods to understand diversity much more. And so what I wanted to do is just end with a few examples that have been published recently that that show different ways of approaching partnerships. So here's an article that was published in 2023 about researchers networking in Mexico. And the research teams reported that they meet with political, religious, and community leaders, communicate the nature of the study, answer all questions, and engage with the community. There's an emphasis on indigenous and rural communities to ensure understanding of the study. There's joint analysis of the data with Mexican researchers and trainees. There's a promotion of local leadership. This is another article published in 2019, Facing Up to Genomic Justice. There's an aspect of education where a researcher talked about ancestry testing she's doing in Afro-Mexican communities, in communities where people traditionally have not thought about or acknowledged their African heritage. And she's performing ancestry testing as part of that education. There's a discussion about benefit sharing. What does benefit sharing look like? Does that mean that pharmaceutical drugs that are developed from, say, a clinical trial will be available? Will there be educational programs? Some might ask about other resources like contribution to hospitals or libraries. Does benefit mean discussing research results with the community? Does it, what about community consultation, discussion to ensure that manuscripts do not harm the communities? There was a researcher here who talked about African countries that have experienced civil war. And when you've been taking data, using data from those communities and reporting it in a way that's sensitive and considers the recent social history that's going on in that African country. These are all just examples that I'm excited there's some conversation about as we talk about understanding ancestry. Because if we want to engage some of the most genomically diverse groups that we know the least about, it will take new approaches and methods to do so in a way to overcome historical issues around trustworthiness and equity. So my concluding thoughts are that racial categories and broad ancestry labels obscure understanding and discourse about underlying genetic variation within populations. The path to understanding global genomic ancestry has been narrow, uneven, and uncertain. How do we widen that path so more scientific leaders can take part? Equitable partnerships with diverse investigators and communities, along with scientific innovation, is necessary for sustainable, trustworthy global genomics research and discovery. And so we spent a lot of time today talking about deficiencies and, or not deficiencies, but opinions and how socially we think about race. And one person asked me once, well, are you saying we should erase race? That's not what I'm saying. I'm saying socially it's not important. But if we want to get to a point where we really can have these conversations about ancestry, we have to do the research. We have to get the data and do so in a trustworthy way. So thank you very much.
Thanks for talking, Dr. Kelly. Let's talk about when we get to the discussion in a few minutes. But before we get there, our next talk is by Dr. Barbara Chaiyachati, who's a faculty member at the Policy Lab in the Children's Hospital in Philadelphia. So our talk is entitled Choose from the Options Below, which is like the mystery title. I'm looking forward to hearing what follows.
And I will do the same.
There we go. Thank you all so much for inviting me today. This talk, Choose from the Options Below, I admit I am presenting with some trepidation. I am nervous after all of the amazing content this morning, and I think I've I would have said different things if I knew, but at the same time, anyway, it has really been an amazing, amazing day. And thank you all for choosing to spend the time here today to talk about this. This is not easy. This is not simple, but this is so necessary in order to continue to make progress, especially in an interdisciplinary way. And so when I received the invitation from Dr. Rupp, she very kindly, I said, what do you want me to talk about? And she said, talk about, you're a clinician. You're coming at this from a different space. And so I may use words that I don't know the definitions of and that I may be using differently. And I hope that that opens up our conversation to continue to advance as we all, again, come to this common space. I do have some fortunate receipt of funding, but not related necessarily to the research or work I'm presenting today. And I want to start with spaces of gratitude. So I am coming from this work built upon and with amazing colleagues who have worked with me. I will be sharing some work that I've completed with Dr. Diana Montoya Williams, Michelle Marie Pena, as well as with Dr. Zagawu, Duncan, and Ellison. Additionally, some preliminary work from with the Center for Applied Genomics and Jamie Catalano and Jennifer Hsu, and really my community, my friends, and my family. And that is because I come here not only as in multiple roles simultaneously. So I am a researcher. I have the fortune of having most of my time dedicated to performing research. And the research that I do is translational research. So I do research trying to understand how adverse experiences may become biologically embedded and influence health trajectories over time. One way that that can happen is through epigenetics and the differences of methylation on the backbone of DNA. But we are talking much of today about how DNA sequences vary between ancestry or genetic similarity groups. And I am also interested in socially lived experiences. And those social lived experiences are influenced by the social construct of race. And so this intersection is absolutely something that I face on a regular basis in every research study that I do. I'm also a clinician. I'm a pediatrician. I have families present to me in some of their worst times, and I do my best to serve and support them. And I recognize they come to me with a lot of things around them. And I interface with the medical record, and I look at medical record outcomes, and we know that there are differences by health outcomes when we look at health outcomes from medical record reported race. And I'm also a person, as we all are. And I'm a person in the US, which means that as a person, in 2020, there were some things that happened. And one of those things that happened was the census. I did not expect that the census would be talked about so much this morning. And in my experience of the census, when I sat down at my countertop in 2020 to fill it out, it was the first time that I remembered sitting to fill out the census. And I was suddenly faced with this piece of data that I know is so important for so many reasons. And I am filling out not only who I am and contributing to the perception of me in my community, but who is my partner and who are my children? And how is the racial and ethnic sort of compilation of my household and my family reflected within this document? And who am I as one caregiver of my children, the person to decide which box or boxes to check for my children? And so I remember calling my partner and saying, how do you want me to fill this out for you? And what should we put for our kids? And so what was my role? But I'm still a researcher. And so this also led me into wondering, what is the data that we know about this? And what is the data that we can think about in terms of race in census over time? And this paper from the working paper from census.gov really reflects how some of the assumptions that we may make, although I'm sure you guys don't, are not necessarily fair. So, right, when we look at this paper overall, we see that 124 million people identified as white alone in the year 2000. That number is approximately the same in the year 2010. But when you link at the individual level and actually look at who checked what box in 2000 and who checked what box in 2010, 2 million people left the category, 1.4 million people came into the category. And so while for the population who was white alone non-Hispanic, that meant that 97% of people were still in the category. For groups that are less frequent, that gray box in the middle is a much smaller percentage of the total population than for the white alone non-Hispanic population. And overall, they estimated that 6% of all linked responses had change, with 9.5% of children having a change in their demographic race group from 2000 to 2010. And we know that changes are happening. So the 2020 census, my family contributed to this increased rate of multiracial identities with a 276% increase as people are recognizing our own and our family heritages and boxes as more complicated. And of note, some other race is now second most common selection across the American population. And so in that context, in my role as a mother, as a clinician, and as a researcher, it led me to this question, or to this statement of choose from the options below, which really reflects that while we all, or many of us, use race and ethnicity data in health research, many times we are using secondary research of clinical data. And while that absolutely has important insights and is really a cornerstone of access and utilization evaluations in health administration data, it is very much imperfect. And so I had the opportunity to sit with some colleagues and really chew through this and think about what does this mean for us. And I couldn't come up with a better way to say it, so I'm going to read a really long quote. As mothers of children with multiple racial and ethnic identities, a prompt that could take just moments sometimes becomes a weighted pause, reminding us of the intersectional reality of our families. Selecting every possibly relevant box might imply that our children are counted as their whole selves, described by each part within. But do we also try to anticipate their eventual self-determined identity that will coalesce with their socially assigned one, as both may contribute to their health status? Or we can reflex to other, recognizing that any collection of small boxes will never equal the sum of their inherited legacies and gathered experiences. A critical acknowledgement as we seek to disentangle the drivers of disparate health outcomes. But if we designate our children as other, how will they be accurately ascribed risk in clinical practice or research? And we sort of came up with some broad general action items, one of which was to increase data transparency. Let's just think about the clinical research we're conducting, and let's try to increase the transparency of the variables and the data decisions and the choice architectures upon which we are building our approaches. So we need to review our assumptions and disclose the decisions that we're making about aggregating and disaggregating groups across choice architectures. And this led to a really interesting question for me as a researcher of what is the documented race change in the pediatric medical record? I looked at the census and I found there was actually a substantial amount of change when we looked at the individual level. Is that happening in the medical record? So we pulled every race data point that had ever been collected in our medical record system and looked at the individual level in order to see how often are patient records actually moving between group categories. And what that means is that if you draw your patient sample, if you download your data set today versus you download your data set in 2 weeks, how many people are actually going to be changing categories across that time? And so overall, we saw almost 7% of bodies change their category at least one time. With the really important point that I'm talking about pediatric records. So to go back to my role as a person, this is really just 6.7% of responses are changed. I don't know how that actual body of a child is experiencing or changing their identity of race. This is just the value that's in the system. So this is a composite value of caregiver reported race, of like administrative, someone has checked in, let me check a box race, of many other forces. But it is changing in a fair number of cases. 6.7% overall. And when we stratified by encounter count, right? So it's unlikely, it's less likely that you will have a change if you've only had 2 encounters in the medical system, as opposed to more encounters in the medical system, was what we would predict if this were truly something that is changing in a somewhat thoughtful way and not just by random someone picking the wrong box. And that is also what we saw. So in people who have in patient records with few encounters or 1 to 4, there was overall 4.1%, up to 8.5% when we looked at those who had more than 16 encounters. And we were also able to say, what are the patterns of change? So this is a depiction of every pairwise change in race category within the system. And the most around the circle, although it may be hard to see, are the different categories that are available in the medical record. And around the most common category change, the most common pairwise combination, are records changing from the other category to the white category. But really almost every other option is reflected. I say almost because I think there's one technically that wasn't reflected, so almost. And just to assure you that this is not just CHOP, more papers have come out subsequent to this. So this is a paper that was just published a couple weeks ago in JAMA Network Open, showing that, and this is limiting to only people who had 2 encounters. So this is a registry of patients who had at least 2 encounters in an emergency room in the state of Michigan, where this data set came from, I believe. And in 2 encounters, 1.7% of adults with 2 encounters had a change in race record, and 7% of children had a change in race record across 2 encounters. But again, I said all of this data is this administrative data, which is going to reflect all kinds of different sources and data. And so this paper, published by Hong et al. Last summer in the Journal of Hospital Medicine, did a survey where they just took a survey into patient rooms and said, here are the OMB categories for race and ethnicity. What is your child? How do you classify your child? And then they looked at the medical record. And when they did that real-time comparison, they found that there was approximately a 30% rate of disagreement for race category and a 20% disagreement rate for ethnicity category. And importantly, more than 1 in 5 caregivers reported that those OMB categories were poorly descriptive of their children. So when we actively solicit parent-reported race versus EHR documentation, we see this substantial difference. All of this is also dependent upon the category choices. So I mentioned OMB. They reference the 1997 OMB guidance, which has what many of us have, here's the Federal Register, what many of us are accustomed to with race and ethnicity as 2 separate questions, recognizing that race and ethnicity are both constructs, which in medical research are sometimes intercalated or conflated. And that is all built upon the 1997. And just as I'm sure you all are aware, but we have new guidance that is coming out. And so while you all are absolutely aware of and appreciate the need for thoughtful and careful integration of what is the social construct of these values, what are we asking people, what are researchers using, the 2024 guidance with the sort of collapse or flattening, I appreciated that word earlier, the flattening of race and ethnicity into one question, I think is going to be a substantial change. And I don't know, I cannot predict what is my patient demographic panel going to look like in 2028 when my patient panel is now going to be responding to a different set of questions. And consistent with what you all, with the overarching hypothesis of today, is to emphasize that it is reflected in the OMB criteria that defining race and ethnicity is set forth as sociopolitical constructs and are not an attempt to define race and ethnicity biologically or genetically. Okay, brief intermission. Pause, a little bit about me. I grew up in the Midwest. I always wanted to be a doctor. So I went to college. What was my major?
Biology.
Biology. Exactly, right? So then I was actually biology, right? I do medical research. So to bring it back to this place of this really important disentangling of race as a biologic construct from genetic similarity as a biologic construct with the lived experience of race as a social construct, the reality is that when we're doing medical research with medical outcomes, many of us, including me, has that short wire connection to say medicine is biology. And so when we look at medical literature and we think about how are we using race in our medical studies, race in medicine absolutely risks reifying it as a biologic construct when we're not using it with intentionality and really thinking about what is actually underpinning it. And so looking at what is race, when we think about medical literature and we see places in medical literature where race is inappropriately conflated and inappropriately used, often words like genetics are invoked. And people inappropriately, but often conflate race is maybe this difference in between race groups is related to some intrinsic genetic underpinning. And this is difficult because there are single cell diseases that when we hold in our mind a picture of who is the most common patient afflicted with this single cell disease, we have very strong associations with race construct pictures in our brain. So as an example, sickle cell trait was present in 1.5% of all births in 2010. And there were differences by newborn screening categorized race groups. And so while you can look at this graph of data that I abstracted from MMWR as saying, okay, children, sickle cell trait is most common among infants who are identified as Black or African American on their newborn screen, that is an accurate statement. What I think is also really important and potentially underappreciated is that those other bars aren't zero. And that we do continue to have, while it is accurate to hold a most common picture, we also need to continually push our quick thinking, medicine is biology, into slow thinking in order to understand that these are not, race is not a biologic construct. These are not closely tied associations. These are complicated and nuanced, population specific, time specific constructs that will continue to change over time and space. And so this led me to another question. As a researcher, what are race and genetic similarity group connection patterns? And this is something that has been brought up and that I think continues to, while we have base data, while there is absolutely data that gives us some information, also there is data demonstrating that this is a continually moving process. And importantly, within pediatrics, I think we have a whole nother set of questions that are not thoroughly, are not as thoroughly investigated. And that when we think again about self-identified race or survey-identified race constructs in pediatrics, we're really only actually thinking about proxy-identified race. And what does this adult caregiver think of this child's race? And that opens up a whole nother question of, do both caregivers think, if there are two caregivers in a family, if there is a third care, does everyone think of this child's race? And would everyone categorize this child's race as the same boxes? And so one more piece of preliminary data is that I wanted to know the answer. So I'm currently looking at a large pediatric population, limiting those to only born after those born in 1980, 65,000 samples, and looking at 3 different sources of race, ethnicity, construct, enrollment survey, medical record, and then genetic similarity as defined by multidimensional scaling to 5 groups. And I am trying to continue to push forward with the language of genetic similarity, because that, from a space of ancestry, I think that it feels accurate to genetic similarity, and that I am not, by looking at the gene sequence, aware of anyone's actual geographic origins, nor do I want to continue the artificial process of othering people to say that people are from not here in any way. And so I do continue to try to use genetic similarity, and I apologize if I don't all the time. So here's the data. So overall, one side is one survey option in terms of categories that may be reflected with, or categories that are reflected within an enrollment survey. The other side are categories that are reflected within the medical record system. And the middle are the 5 MDS groups to which populations may be attributed. And while there are common groups that have some consistency across these three choice architectures or mapping options, it is not perfect. There is a lot of movement, and it creates, in the end, a really beautiful picture, but a beautiful picture that reflects nuance and complication and does not allow us to easily say someone is this because of one thing or the other. Now, data that I'm not showing, but that I think is also really interesting, is that to the point of ancestry, the reality is that when we define genetically similar groups, we are actually doing that, right? We are saying, okay, there were a population of people that the Human Genome Project went out and plucked from a certain geography in the year 2007 or 2010, or whenever that sample was collected. And we are saying, what is your genetic similarity to that population? We are saying nothing about what the geographic history was of that population. We are in fact saying, who is that population right now, then? And how much are you genetically similar? So interestingly, in this data, we have seen, and other data as well, that likelihood of assignment is degrading in real time. Our genetic similarity, the percent likelihood of your MDS assignment to any given category, is statistically significantly degrading over time, because we are continuing to have all of the forces that are current and contextual in US society. And so we are not creating populations of people that are more different. We are creating populations of people that are more the same. As a pediatrician, this is so important, because all of those things are based upon adults. Kids are different, and kids are just continuing to become more and more and more different every year. And so I do all of this because, as the third hat, a clinician, when we stratify groups by populations, health outcomes may be different. And we just all want to know why. And so, in perfectly on time, conclusion, final reflections are: health difference across population groups invites deeper inquiry. While we need to get into the health differences and we need to get all of those places, I would argue that we also need to really make sure that we are starting from the top. What are those groups? How are we defining them? What are the assumptions that we are making about group identification, generalizability, and variability? When we understand more the where and the when of health differences by what those groups actually are, I think we need those things defined and well articulated before we can get to the why those differences exist. And then we can move on to the what next and how do we start to address the deep needs for improved health equity in our society. Thank you.
just gonna have it just so here.
Okay.
Bringing it up.